So we have finished with we have established the fact that we have normally a osmotic filtrate that enters nephron loop right here. It becomes concentrated first. This is why it is called multiplier. It increases the concentration as it goes down. Okay. Then reabsorption of sodium decreases the concentration, and then essentially what regulates the osmolality of the urine? Essentially presence or absence of ADH. Does that make sense? So actually it's kind of worthwhile to introduce the concept of obligatory versus facultative reabsorption. The word obligatory, what does that mean? If something is obligatory, yeah, it's necessary. It must happen, right? Facultative, it doesn't have to. So essentially, if you think about the water reabsorption in the nephron loop, is it obligatory? Does it have to happen? I'm asking simple questions here. What do you think? Did I say anything about like any hormones, any regulations? No, it happens. That's it. It's obligatory. It must happen. Does that make sense? So this is obligatory water reabsorption. This part, it's the facultative one. There's ADH, there's reabsorption, there's no ADH, there's no reabsorption. Make sense? So on the left, basically, you have sedation when you are overhydrated. You are overhydrated. Osmolality of plasma extracellular fluids is really low. Chemoreceptors in hypothalamus sense it, do not send the signal to produce ADH. There's no ADH production. No aquaporins inserted into the collecting duct. No water reabsorption in the collecting duct. You pass a large volume of diluted urine. Does that make sense? If you have the opposite situation, when osmolality increases due to the dehydration, ADH levels increase, okay? Number of aquaporins increases. You've got yourself rehydration. Nuclear reabsorption of water. Now, what is the role of urea? It turns out that urea is necessary to maintain that gradient, because I actually never mentioned what forms this gradient in the kidneys. When we talked about osmolality, let me ask you again, what is osmolality? It's a number of particles. So say if you have solution of sodium chloride and potassium chloride, they are chemically different, but the osmolality can be the same if you have the same amount of particles. Does that make sense? So that's the same. Urea will be just as osmotic as anything else. And it turns out that urea maintains that gradient that we see from 300 to 1200 from cortex to medulla. That makes sense? So here's what happens. As the filtrate passes through the nephron loop, here in the beginning of the ascending limb, urea starts to be absorbed. Well, should I say, no, it's not absorption. Starts to get secreted into the ascending limb, right here, okay? So at this point, concentration of urea in the filtrate is relatively high, okay? As it enters collecting duct, due to the water reabsorption, urea concentration increases even more, and more, and more, and eventually urea diffuses into the inner medulla, maintaining that higher concentration of urea in the inner medulla. Essentially what happens, water reabsorption increases urea concentration, which allows urea to diffuse and maintain the higher osmolality in the deeper medulla. Does that make sense? And urea is constantly brought into the filtrate as the product of protein metabolism. It's a normal nitrogenous substance, the product of oxidation of proteins. That makes sense? So it turns out that urea that we excrete with urine plays an important physiological role in kidneys. And by the way, that's one of the reasons, if you, as far as I know, if you need to cook kidneys, what's the first thing that you need to do? You need to put them in water. So water will basically extract all the urea out of kidneys. You still can cook them, it's fine. When people say it's like a cooking a filter, that's not entirely true, because as you now know, nothing stays in the kidneys. Does that make sense? Now, I want to give you examples of some of the diuretics here. Kind of, try to figure out mechanisms. Why do we need diuretics? What diuretics are used to treat? So congestion, systemic congestion, or you kind of taking it to the extreme before high blood pressure. And actually, the number of clinical studies show that that's the most effective way to treat the blood pressure. It's kind of interesting. Beta blockers, not as effective. Diuretics are the best. So what you can do, you can inhibit sodium-potassium pump. Make sense? That's kind of a lousy though, because you need something to leave sodium in the filtrate. Sodium-potassium pump, sodium channels inhibitors, you can do that. There are so-called loop diuretics. They specifically inhibit the reabsorption, water reabsorption, and sodium chloride reabsorption in the loop. There's a very specific class of diuretics that inhibit aldosterone receptors. When there's no, there is aldosterone, but when aldosterone is inhibited, sodium is not retained, right? Water is not retained. There's diuresis. And finally, there are diuretics that inhibit ADH receptors. So they actually influence not the obligatory, but facultative reabsorption in the collecting duct. That makes sense? There are multiple classes of diuretics. Now, let's talk about the kidney function. It's a renal clearance, and it's quite fascinating. So this is the parameter that is used by clinicians to assess how kidneys work and whether a patient has renal disease or not. Okay? So what is the renal clearance? How is it calculated? It's the concentration of substance in the urine multiplied by the rate of urine formation divided by the concentration of substance in the plasma. So the amount of urine produced in a minute is measured. Concentration of substance in the urine is measured. Concentration of substance in plasma is measured. And if you would take these parameters for the drug called inulin, you will find out that renal clearance for inulin is 125 milliliters per minute. Now it's all fairly vague when I refer to it in numbers. Let me explain what renal clearance physiologically means. Renal clearance reflects whether the substance is completely cleared out of the blood or not, and all the degrees of that process in between. Does that make sense? Imagine you have a substance like inulin that is completely filtered and not reabsorbed at all. Do you understand what I'm saying? So everything that is filtered is released in urine, is excreted in urine. Does that make sense? For substances like this, renal clearance equals GFR. Free filtration. Renal clearance equals GFR. Now, inulin can be used, it's a carbohydrate, the plant carbohydrate, which is not toxic and can be used as an external marker to evaluate glomerular filtration rate in people. You basically can feed a person with inulin, take a blood sample, take a urine sample. Does that make sense? Easier way to calculate renal clearance, sort of faster, is to measure creatinine in the blood and in the urine. Turns out that creatinine, which is the product of the dephosphorylation of creatine phosphate in muscles, creatinine is freely filtered. For creatinine, renal clearance is basically equals GFR. There is no reabsorption, there is no secretion. Does that make sense? Are we clear about it? So you can measure creatinine in the urine, you can measure creatinine in the plasma, you can measure the flow, the urine rate formation, and you can calculate GFR. And if it's, now, is it truly like 0,0? No, of course. So if you use creatinine as a marker and get GFR of 123, it's fine. If it's 126, it's fine too. It's a range. It's not 125 or dead. Does that make sense? If the substance, now think about the term renal clearance. If renal clearance of a substance is 0, what does that mean? Is it filtered in the very first place? Sugar, is it filtered? Glucose? Yes. Is it fully reabsorbed? Yes. So there's nothing in the urine. Does that make sense? Renal clearance is 0. It's filtered and then completely reabsorbed. Does that make sense? So I kind of give you two opposites here. One, no reabsorption, no secretion, free filtration. Things like creatinine or inulin. Does that make sense? Another opposite, filtration and then complete reabsorption. Are we clear? So that's renal clearance 0. Now, if renal clearance is less than GFR, means that some of the substance is reabsorbed. Does that make sense? The more substance is reabsorbed, the lower its renal clearance. Is that clear? No pun intended. Do I understand that? There are situations for some drugs, like penicillin for instance, when renal clearance is higher than GFR. It means that the compound is not only filtered, it is secreted on top of it. Does that make sense? Do you understand what I said? You got it? I'm going to ask you questions about this. I'm going to give you an example of a compound. I will give you its renal clearance, and I will ask you what it means. Is it fully reabsorbed? Is it partially reabsorbed? Is it freely filtered? Is that filtered and secreted on top of that? Do we understand that? Now, why it is so important to measure GFR? Because we can assess kidney function. Does anyone know what is the clinical threshold for chronic kidney disease? I'm not going to ask numbers. I told you before. It's 60. Basically, when we have a patient with a GFR in the range between 90 and 60, it's a high risk for chronic kidney disease. GFR of 60 and below, it's a chronic kidney disease, and I think GFR below 30 is renal failure. There are more narrow gradients. Basically, each range of GFR suggests what to do with the patient. Like, say, above 90 but below 125, observe maybe, lifestyle changes. Below 90, observe tightly, definitely lifestyle changes, because high risk of chronic kidney disease, low salt diet, things like decreased blood pressure, things like that. If GFR becomes 60, then it's a problem. Okay, and I think 30 is the dialysis. Okay. Or kidney transplant, which is, by the way, totally doable. Yeah, the only problem is to find a match. Now, let's talk about role of kidneys in homeostatic regulations. First, water balance. It's kind of boring. Not boring, actually, it's kind of fascinating. So this little scheme on the left shows you the intake and output of water during the day. So this is how much we drink. This is how much we get from food. So about 2.25 of a liter. It varies greatly, but it shows you sort of a normal metabolic homeostatic rate of water intake. Does that make sense? So can you drink a gallon of water a day? Absolutely. Should you be away from the toilet for more than 10 minutes? Probably not. Okay? Make sense? Now, how do we lose it? The majority of it we lose with urine, which is regulated. Some of it we lose by skin and lungs. Now, skin is regulated. You can sweat more or less, but it mostly depends on the temperature. So think about this. And feces and sweat, okay? And well, sweat part of the skin. So skin loss with the sweat, loss with breathing, loss with feces. Feces do not contribute much. Water reabsorption in large intestine is practically all obligatory. So there's not much regulation, but it's only about 4%, so nobody cares. Okay? That makes sense. Sweat. In this case, the regulation is not about water. The regulation is about temperature. Does that make sense? So it's more important to maintain body temperature than to take care of water at this point. Now, urine, or loss of water by urine, the objective here is to regulate the water balance, actually. Do you understand? So we have like, for sweating, the objective is not water balance, the objective is temperature balance. For urine, the objective is water balance. It has nothing to do with temperature. Okay? So, what happens if we, normally, hunters and gatherers, humans, are they overhydrated? Never. Right? So, like, why I'm drinking this? Why I'm drinking tea? I'm drinking tea not because I'm really, really thirsty, because my mouth gets dry when I'm talking, you know? That's it. So I'm actually overhydrating myself compared to my actual needs. Now, actual problem is dehydration. In dehydration, osmolality of extracellular fluid increases, which actually decreases the production of saliva and leads to dry mouth. When you have dry mouth, what do? Drink. That's it. Okay. Essentially, you are becoming thirsty. That's it. That's actually quite elegant. Make sense? Plasma volume. When you dehydrate it, volume of plasma goes down. What happens to your blood pressure? It decreases. What happens to granular cells in kidneys? What do they release? Renin. Renin activates angiotensin-aldosterone system. Aldosterone is released. One of the mechanisms is stimulation of thirst. Another mechanism is retention of sodium. Retention of sodium, retention of water. That makes sense. And this is via ADH, okay? So we've already talked about it. When osmolality of your extracellular fluid, essentially osmolality of plasma, goes up, and it's more concentrated, ADH is released in two effects. Thirst, which is not shown here, but it does stimulate thirst. And second, increased water reabsorption, which leads to concentrated urine in small volume. Do you understand this mechanism? We talked about them so many times. It's more of a reminder. Now, sodium. Main mechanism, of course, is aldosterone. So there are two stimuli. Concentration of potassium in extracellular fluid and decreased content of sodium. That's actually quite interesting. Try to concentration of calcium in the blood can change dramatically. Remember we talked about PTH, parathyroid hormone, how it regulates calcium in the blood in the very beginning? Okay, so calcium can actually decrease or increase, and it'll take some time for parathyroid hormone to bring everything back to normal. Okay, now concentration of sodium in the blood or in extracellular fluid in general, concentration doesn't really change. What changes is the total content of sodium. So I'm going to work our way through the left flow chart, and then I'll give you the real life example. So if total content of sodium decreases in the body, this stimulates immediately release of aldosterone. That's mineralocorticoid. It responds to low sodium content. Aldosterone increases reabsorption of potassium, and brings back reabsorption of sodium, increases reabsorption of sodium, and brings back sodium levels to normal. Does that make sense? Now, so I said sodium content is low. When you reabsorb sodium, what happens to the sodium content? It increases or decreases? It increases. Water follows sodium. When you reabsorb sodium, you reabsorb water. So sodium content increases, but concentration doesn't change because you bring water along with it. Does that make sense? Do you like KFC? There's nothing wrong with admitting it. I saw a bunch of doctors, including endocrinologists, ER, and neurologists, ordering like 2 buckets of KFC. It was delicious, honestly. It is. Okay. Few years ago, quite a few years ago, KFC put in the market an absolutely stunning sandwich. You probably know what I'm talking about. They substituted buns with fried chicken. They had 2 gigantic pieces of fried chicken and something like bacon, lettuce, and tomato between them. The hallmark feature of this sandwich, which I don't think they advertised, but it got out, really, was that it contained either 2 or 3 daily doses of sodium that are required. One sandwich. So basically, you're like eating salt with a spoon. And now that's kind of one of my colleagues. I call him my colleague, but he's a professor at the time. So we had a conversation about regulation of blood pressure and everything, and he said, out of curiosity, he said, I tried this. So he bought this thing, and he ate the whole sandwich. He felt blood pressure spiking. Because here's what happens. When you eat this sandwich, sodium enters what? Extracellular fluid. So its content immediately increases. There are 2 sources of water there. Well, 3 sources of water that are available. One is to drink. He didn't. At least not right away. 2, water reabsorption in kidneys. Basically, direct water reabsorption. Because all that sodium draws the water in. And 2, drawing water out of the cells. Since sodium content is what's regulated, not the concentration, the volume of his extracellular fluid immediately spiked, giving him an appreciable spike in the blood pressure. Like he said, I felt it. I felt my blood pressure spiking without any measurements or anything. So, and that's actually one of the reasons why you feel thirsty when you eat something salty. Make sense? And for me, it's terrible because one of the staples, hallmarks of Russian cuisine is salty herring. And I love it. And I can eat a ton of it. And then I drink a huge amount of fluid. And then I need to know where the closest restroom is for the next 24 hours. So that's one mechanism. Now, what if blood pressure increases? One of the mechanisms of regulating blood pressure is through the release of atrial natriuretic peptide. Remember we talked about it? So ANP has several effects. One is decreased renin release. When renin goes down, aldosterone goes down. When aldosterone goes down, sodium reabsorption goes down. Now, when I say sodium reabsorption goes down as a result of atrial natriuretic peptide, when sodium reabsorption goes down, where does sodium stay? Say again? In the filtrate, in the urine. Does that make sense? Now, if sodium stays in the filtrate, how does it affect water? It follows it. It stays in the filtrate as well. So essentially, atrial natriuretic peptide makes you excrete sodium and excrete water, which reduces blood pressure immediately. So you can see that it's not an ultimate goal to excrete sodium or reabsorb sodium. Essentially, sodium reabsorption or sodium excretion are tied to the blood pressure regulation. If blood pressure is low, then sodium reabsorption, which is followed by reabsorption of water, okay, is the way to bring up blood pressure. Does that make sense? If blood pressure is high, then sodium excretion is the way to decrease the blood pressure. Does that make sense? So essentially, this process, sodium is not of a particular value by itself. Good? Now, potassium. With potassium, there's a little problem. Not really a problem, but how is potassium distributed between extracellular and intracellular fluid? Where do you have more potassium? In the cells or out of the cells? In the cells. So if you eat 5 pounds of bananas, which contain a lot of potassium, technically your potassium concentration in extracellular fluid should spike right away. Spike a lot. And it has all kinds of profound effects on the cardiovascular system, nerve transmission, so on and so forth. Basically, it will just mess up all membrane potential thing in all your cells, because potassium is one of the key ions to regulate the membrane potential. The fact that it's higher inside of the cell than outside. That makes sense. So essentially, with potassium, the goal is to maintain potassium concentration in extracellular fluid low. It is achieved by 2 major mechanisms. One is the direct excretion of potassium. You eat a lot of potassium, it is filtered in the glomeruli, and it is excreted with urine. That makes sense. Second step, aldosterone. If we go back for 1 second, elevated potassium concentration in extracellular fluid triggers aldosterone release, which leads to potassium secretion. Does that make sense? I had a graph somewhere. It shows something like that. I will try to draw it from my memory. So if you would, first of all, if you would draw, like, this is the potassium in the extracellular fluid, the more potassium you have, the higher aldosterone levels you can observe. That's aldosterone. Does that make sense? Now, on top of that, if you will try to measure not aldosterone, but potassium in urine, it will also be something like that. Okay? So more potassium you have in extracellular fluid, more potassium you have in the urine. And actually, you can see if you will make somehow, say, animals that lack receptor for aldosterone. So without the receptor, it would look something like this. So you still excrete potassium, but not as effective, because the only mechanism that you have is this one, aldosterone-independent mechanism of potassium excretion. Do you understand what I'm saying? So kidneys regulate potassium, and the main goal here is to get rid of it. Now, calcium. Hypercalcemia. Let's get it out of the way. Hypercalcemia. If you are otherwise healthy person, no underlying conditions, no endocrine pathologies, and you somehow manage to eat 2 pounds of chalk, okay, your calcium levels in the blood will spike. What's going to happen? How do you get rid of calcium? Simpler. It's in the kidneys. The whole thing is in the kidneys. How do you immediately get rid of calcium? Yeah, just filter it out. That's it. It goes away with the filtering. Does that make sense? Everything in excess will be filtered out because there's certain transport maximum for calcium reabsorption in the kidneys. And transport maximum, this thing, you know, the capacity of kidneys to reabsorb, it isn't something randomly selected by Mother Nature. As I always say, you know, people who had different transport maximum all died because we are evolutionary better. We survived and they didn't. So it's actually, you know, it allows us to regulate to maintain proper calcium levels in the blood. So hypercalcemia, temporary hypercalcemia in a healthy person, isn't much of a concern. Now, hypocalcemia, say nutritional hypocalcemia, how will it be compensated? If I say nutritional, that we can pretty much cross out the right one, right? Because if it's nutritional, we don't take enough calcium with the food. So we have 2 more mechanisms left. One, take it from the bones, but we're not studying skeletal physiology here, so screw the bones. Reabsorption of calcium in kidney tubules. Essentially, parathyroid hormone increases the number of sodium-calcium symporters that bring calcium back into the blood in the kidney tubules. Does that make sense? So basically it's PTH. For calcium it's easy. It's PTH that increases calcium reabsorption in the kidneys. Does that make sense? Now, buffer systems and the acid-base balance. We're going to start to introduce this. What is more common, acidosis or alkalosis? Acidosis, yes. I mean, I don't want to be dismissive to people that say, well, look, think about this. What is the problem? What if somebody has a kidney disease? What is the problem with kidney? It can't filter. So it messes up blood pH, and acidosis can stem from conditions like diabetes. So if kidney is messed up, hydrogen ions are not secreted, hydrogen ions accumulate in the blood. That's acidosis, right? So kidney is really important to get rid of those hydrogens. Okay? Now, do you know of anyone with a lung problem that manifests as hugely elevated ventilation rate? I personally don't. Usually people have trouble breathing. If it's COPD, if it's asthma, Does that make sense? Things like that. If your breathing is sort of, if you're short of breath, how does it affect your blood pH? Decreases. It becomes more acidic because you cannot expel CO2 as effectively, and blood pH goes down. So on both sides, on the respiratory side and on the kidney side, the problem is mostly acidosis. Okay? So normal pH of the blood is 7.4. And deviation by 0.05 units of pH, which is, if you worked in the lab and you worked with pH meter, when I do pH of my buffer and it's 7.45, and it's not some really sophisticated, fine biological experiment with some fragile experimental system, I don't give a shit, really. 7.45, 7.4, who cares? I'll put it out there, like in the air. It's going to change in 30 minutes. Here, anything deviation more than 0.05 units of pH is considered to be either acidosis or alkalosis. So it's really tightly regulated. Moreover, it's really hard to change because there are buffer systems. So this is the main buffer system in the blood. It's bicarbonate, right? If you have more hydrogen ions than needed, say if you have like temperacidosis, say you have exercised, vigorously exercised, so you have a lot of lactic acid, okay, produced. Those extra hydrogens will bind to bicarbonate. If you, for some reason, developed alkalosis, say you decided to hyperventilate, then hydrogen ions will be produced by the breakdown of carbonic acid. Ketone bodies can contribute to acidosis. Protein diet generally makes you a little bit acidic. Now, buffer systems, when you say buffer systems regulate hydrogen ion concentrations, they buffer. So buffer systems are great for adjusting to small fluctuations. Does that make sense? Like, you decide to start completely new life from Monday, you switch to high protein diet, your blood becomes slightly more acidic, your buffer systems immediately compensate for it, so you don't really notice anything. That makes sense? Now, respiratory controls will take over also in a short-term perspective. If you develop acidosis, your breathing rate will increase. But the most important regulatory system are kidneys. Okay? It will take them a while to adapt to this, but they are the best in regulating blood pH. Now, what are 3 buffer systems that you're going to find? I'm going to talk about this and wrap it up. First is bicarbonate. It's in the blood. Same, by the way, same buffer system that maintains pH of the ocean. Bicarbonate buffer. Any buffer is the mixture of weak something, like weak acid and strong base, or vice versa. So here we have really weak acid. If I would ever write carbonic acid in the solution as one molecule in the chemistry department, I would get an F right away. Just go home and study. Because from chemical standpoint, that's BS. It doesn't exist in this form. Okay? And bicarbonate. Bicarbonate is the medium to strong base. Okay? So these buffers, extracellular fluid, blood and interstitial fluid. That's the most important buffer in ECF. Actually, the only significant buffer. Okay? Phosphate. urine and intracellular fluid. It's phosphate. So in this case, phosphates, it's going to be what? Phosphoric acid is fairly weak. Actually, it's not so much phosphoric acid, it's ions. So this is dihydrophosphate, and this is hydrophosphate. So these two ions can either accept hydrogen or donate hydrogen. This is more acidic, so it's readily giving hydrogen away if needed. This is more basic, so it accepts hydrogen if needed. Make sense? Actually, if you ever read the content or ingredients of Diet Coke or Fanta or something like this, you're going to find as one ingredient phosphoric acid. It sounds really horrible, but phosphoric acid is used as a buffer system in those drinks. And yeah, it makes them definitely acidic. Finally, proteins. Remember when we were talking about respiratory system and transport of carbon dioxide in the blood? We mentioned that this hydrogen here is buffered by albumin or hemoglobin in the red blood cells. So these are most important in plasma, which is absolutely logical. There's a lot of protein in the plasma, and inside the cells. Remember, extracellular fluid is really protein poor. Does that make sense? So when pH increases, carboxyl groups of amino acids can release hydrogen. When pH decreases, amino groups can bind hydrogen. Does that make sense? So we consider this all about hydrogen. If something is acidic, it means there's a lot of hydrogen ions. If something is basic, it means there's very little hydrogen ions. We're clear? So we're going to wrap it up